frequency, basically the Hardy-Weinberg equation it deals with population genetics. So basically, evolution happens in a population. So just to kind of, I know evolution is kind of a polarizing topic to a lot of people, and this isn't specifying whether it's micro or macro, but I'll go ahead and kind of talk about that. Microevolution is change within a species. So basically, like if you even look back at humans in 1950, you know, they were shorter in stature, they were less, I guess, less muscle proportion and that kind of thing, and that's just over, you know, 60, 70 years. So you go back even further, you know, thousands of years, and there's differences, little minor differences, but it's still a human. So that's what microevolution is talking about. And then macroevolution is saying like, there was a common ancestor that was a bacteria a long time ago, and that every species came from macroevolution. And this isn't really going into either one of them. It's really probably describing microevolution more because it's just kind of how it occurs. And basically, when you have a population of people, if you want to measure, basically Hardy-Weinberg keeps everything stagnant. So it's like, if you know something, you can look it up and make calculations on it, and if that changes, then you know evolution's happening, and you can kind of see how fast it's happening based on the baseline. That's why he was saying everything that drives evolution, you just kind of ignore that. So the purpose of it is to see how fast evolution is happening in a certain population, and that's how, you know, all goes back to ultimately gene frequency, which is alleles. And this is from last test, but I want to make sure everybody knows what this is. So what we're going to do is we're going to dissect these two equations and see how they interact with each other, and also just kind of apply it to one principle here, and we'll just dive into it from every perspective, so hopefully that the homework will go pretty clear. But let's do cystic fibrosis. That'll be what we're looking at in a population. So cystic fibrosis is a Mendelian genetic trait, which means it has, you either have the healthy allele or you don't. You have the diseased allele. So it's one of two ways. It's on chromosome number 7. And so with parents, a lot of times you have two parents typically. And so we'll put 2 chromosome number 7s here. And there's a gene just kind of randomly somewhere on the chromosome. Well, it's not random, it has a specific place, but I don't know exactly where it's at. That is basically the cystic fibrosis gene. And so CFTR is the name of the gene, but we're just going to call it the cystic fibrosis gene. And let's say that this is the dominant, that this one's dominant, so it's going to be a capital C. And if this one was recessive, this would be a lowercase c. So that's going to be our dominant trait and our recessive trait moving forward. So in this particular population, basically this equation tells you how many alleles. Like it's telling how many of these there are and how many of those there are. And so if you had, to simplify it, let's just say you had a bucket of 100 people or a bucket. It's hard to put 100 people in a bucket, but if you had a bucket and you had pieces of paper that had one of these 2 alleles on it, if you just randomly picked out a piece of paper, what's the probability that it would be this one, or what's the probability it'd be this one? That's what a gene frequency or alleles is talking about. And then the genotypes is in so if you know anything about kind of probability in math, if you're going to try to see, if you had this bucket of pieces of paper and you had a certain number of dominant alleles and a certain number of recessive alleles, let's say that you take, what's the chances you could get 2 dominants in a row? And that's basically that times itself, which is p squared. So p squared represents homozygous, because that's 2 dominants, dominant. So that, and you just basically just need to memorize these. They're not that hard. There's 6 questions on the test out of 50 that relate to this. And then there's a 30 out of 30, 30-point homework assignment that you have over the weekend for lab. So it's just best just to kind of get familiar with these enough that you know that P squared, that P represents the dominant allele. And that P squared is, if you were to pull that allele out of a bucket randomly twice in a row, what's the chances? It would be whatever that number is out of 100 times itself, and that gives you P squared. So we'll actually put numbers to it, and that'll make more sense in a minute, but I just want to kind of get the concepts down first so that you know what these P's and Q's and all that are. So if P is the dominant allele, what do you think is that Q represents? Yeah, the recessive allele. And so, if Q represents the recessive allele, and we only have 2 types of alleles, you either have the disease-carrying gene or you don't. So there's only 2 different types of alleles. The dominant one is the healthy gene, and the recessive one. So cystic fibrosis is a homozygous recessive disorder, which means that to have the phenotype, to have the disease, you have to have 2 of these. So basically, that will be what Q squared is. So Q squared, so Q by itself is just the recessive allele, but if we were to get 2 little C's, pull out back to back, we'd have to multiply times itself, and that would give us the genotype, which is homozygous what? Recessive, yeah. Alright. So, if this is homozygous dominant and this is homozygous recessive, what do you think 2PQ stands for? What's the only kind of other combination we could have of alleles?
Heterozygous.
Yeah, heterozygous. And you wouldn't say dominant or recessive because it's just one of each. So you just say heterozygous. And in the Greek or Latin, wherever it comes from, hetero means different. So you have 2 different ones. You have the allele for the dominant allele, and you have the recessive allele. And the reason there's 2 there is if you think about it, you could get one from the mother, one from the father, or vice versa. So you could get it either way. So that's why they have the 2PQ there as far as making the equation. So let's just kind of summarize what we know so far. So the P plus Q equals 1. That's the equation that we use if we know the alleles in a population. If we happen to know the genotypes in a population, we could use this equation where it's P squared plus 2PQ plus Q squared equals 1. And so basically, if you just know, like in some cases, one little piece of information, you can get any of these. For example, if you know homozygous recessive, if you know how many in a population, say there's 9 people out of 100 that have cystic fibrosis, then we know that we automatically know that what the homozygous recessive is, because we know you have to have 2 little C's to get that. And then all we have to do from there is figure out P from Q. So the way we do that is anytime you go backwards, like if we want to figure out what Q is so that we can figure out what P is, you just take the square root. So you would take the square root of Q squared, and that would give you Q. So it depends on what angle you're coming at. And that probably kind of confused you a little bit, and so we'll back up and we'll put some numbers in so you can see how the numbers kind of flow. Let's say that, and also just kind of how to know how to go back and forth from decimal to percentage and understanding that. That's kind of basic, but just to make sure you don't get lost in that. So again, alleles, dominant alleles P, recessive alleles Q. If you get two dominant alleles back to back, that's homozygous dominant. If you get one of each, that's heterozygous, which is always 2PQ. And if you have homozygous recessive, that's two of the recessive alleles. And basically, you don't always have to do the whole equation. You just have to see what the question's asking for. If the question's asking you for how many heterozygous do you have in this population, if you have, say, P, you can find Q because you know you can just subtract it from 1, and then you have P and Q, and you just plug them in here, so all you'd have to do is just use that one little middle part. So you just got to see what it's asking for. You don't have to work it all out every time. And we'll look at it from different angles. So let's say that this is our chromosome number 7, the CFTR gene, which is the cystic fibrosis gene. Let's say that in a population, generally, not a lot of people have cystic fibrosis. So we're going to say that the dominant allele is 0.8 of the population, or 80%. Because if you take a decimal, you multiply it times 100, it'd be 80. So that's 8%, 0.8, because that's how this is structured. We're going to keep it in decimal form. And so if we have P, and we know it's 0.8, we have this equation. We know what Q is, right? What would Q be? Yeah, 0.2, because this has to equal 1. So 0.8 plus 0.2 equals 1. So now we know that Q equals 1 minus 0.8, so Q equals 0.2. So now that we have P and Q, we can figure out anything we want to figure out over here. So we now know that 80% or 0.8 of this population, we'll say it's 100 people, are have the dominant allele. So in this bucket where we have pieces of paper, 8 out of 10 times you'll pick the dominant allele, so the big C, and maybe 2 times out of 10 on average you'll pick the recessive allele. So now we have this, and we can plug P and Q in it over here and get anything we want. So now if the question asks, if the structure of the question just told us that 0.8% of the alleles in a population were dominant for the healthy gene in cystic fibrosis, and it said how many, what percentage, or if it said what percentage of the population has recessive alleles, then you would just say 0.2 or 20%. So you just have to be able to. First of all, you have to know the definitions of what the dominant allele is, the recessive allele, what it goes with, the homozygous dominant, what it goes with, heterozygous, what it goes with, homozygous recessive, what it goes with, and then just basic math, knowing how to square something and how to take the square root of something. So that's what we have to do over here. So now we have P and Q, so just kind of organize it. We have P equals 0.8 and Q equals 0.2, because 0.8 plus 0.2 equals 1. So we have our allele frequency, or our gene frequency. Those are interchangeable because this is a gene. And let's say that we want to find out how much of the population is homozygous dominant. So the chances of, basically this is asking that if we have this bucket of pieces of paper and 80% of them have big C's that are healthy, and 20% of them have little c's that are the disease carriers, if we were to pick 2 of the big C's out in a row, what's our chances of that? Do you think it would be higher or lower than this number of just getting 1? You follow me there? Would you have a greater chance of just getting 1 of these in a row, or 2 of these in a row? You'd have a greater chance of just getting 1, right? So just to kind of follow the math in your head, you know this number is going to be a little bit smaller, or it's going to be smaller than this number because it has to be 2 in a row. So we're going to multiply 0.8 times itself. That's what P, if we want to find what P square is, that's what's homozygous dominant. We have to multiply 0.8 times itself, so that's 0.8 times 0.8. And then p squared is going to equal, if you put 0.8 in the calculator, it'll give you 0.64. So that's smaller, so that would make sense. So your chances of grabbing 2 of these, if 80% of them are the big C's, are about 60% if you were going to get 2 in a row in this population. All right. So let's see what our chances of actually getting the disease. If we were to pull 2 of the disease-causing alleles, the recessive ones, in a row, we know our chance of getting 1 is 0.2. So what's our chance of getting 2 in a row? Is it going to be bigger or smaller? Smaller, yeah. So so here we're looking at for q squared equals, and it's 0.2 times itself. So when q squared equals, and that's 0.04, you can use a calculator on these, so 0.04. So basically 4% of the time you could get 2 little c's in a row if 20% of them out of 100 in this little bucket were little c's. And so in this particular population, only 4% of the population has cystic fibrosis. Does that make sense? Because that's the homozygous recessive. Because the carriers don't have the disease, and definitely the dominants don't have the disease. And the carriers don't have the disease because this is a homozygous recessive disorder. And then if we want to see what there's 2 ways we could see what the heterozygous is. One is we could just take 0.8 and 0.2 and plug them into this equation. And so if we do that, so you have 2pq, and this tells you heterozygous. So 2 times 0.8 times 0.2, that's a 2. So now we have, we can just do 2 times 0.16, which is 0.8 times 0.2, and then this will give you 0.32. So that's just plugging it in and doing the math to see what that is, because we know what the allele frequency is. Or, what's one more way we could do that? A different way, and you can pick whichever one lights your candle. If all this equals 1, and we know this and this, then whatever's left is what that is. So let's try that. So let's say that we know that PQ is 0.64, and then this is what we're solving for, is the middle one. So we'll just make that, we can make it x if you kind of want to set it up like an algebraic equation, plus, and then we know that 0.04 are homozygous recessive, and that equals 1. And so if you isolate x, you have 1 minus, and then you just put everything over there, 0.64 minus 0.04, and then x equals 0.32. Hopefully. Let me make sure it almost doesn't look right. 1 minus 0.64 minus 0.04. Yeah, that's right. So, you know, if you had, basically, you know it equals 1, so if you're missing any of these, you can just kind of set it up that way. It just depends on how you want to do it. If you want to square it, you can, or if you want to do the 2PQ, you can, or if you just want to solve for what's missing, you can. So, everybody, any questions over that? We're not moving on yet. I'm just seeing if you followed that okay. All right. So, now that we're kind of familiar with these numbers and we know kind of where they're going and what they look like, we'll start manipulating this different ways and see what we can find.
So we gotta put X anywhere in that equation?
Just
It
what,
came out.
where you're missing something.
Right.
Yeah.
Would've came out.
Yeah.
Whatever.
Or you could put 2 PQ, but X just kind of makes it cleaner. All right, so let's say that the question asked if a population has, I'm not wording this very nicely, but 4 out of 100 people with cystic fibrosis. How many, let's see, let me word it different. So we know that we know how many people have cystic fibrosis in a population, and then we want to find out, out of 100, how many people are carriers for cystic fibrosis in this population. So see how basically you really have to know the definitions of kind of what homozygous dominant, homozygous recessive, and all that is to be able to interpret a question like this. So we know that a population has 4 out of 100 people with cystic fibrosis. What is that telling us in this equation anywhere? It's giving us a hint. It's giving us the answer to it. Want something up here. We know something up here, either in this equation or this equation. Q? Q squared. Yeah, you're close. So Q squared, because we know that to have cystic fibrosis, it's a homozygous recessive disorder. So we know that you have to have 2 of the little c's to get it. So this is telling us that 0.04 or 4% of this population is homozygous recessive. And this is real world how they use this basically, because they don't know the alleles, and they're not just given the genotypes, but what they know a lot of times is if you have a disease, and a lot of times diseases are recessive, and so if you know a certain disease is homozygous recessive, you have a certain number of people, maybe it's 10,000 people, and you find out that there's 1,000 people in that population that have cystic fibrosis, or then you know that you're homozygous recessive for 10%. So that's how they start a lot of times, is they'll find out, and that's the key. If you can find out homozygous recessive, you can find out anything else. So if you can go that route to start, that's a good start. And you can approach these problems different ways. This one you can't, this is about the only way you can approach this one. So if we want to know how many carriers for cystic fibrosis we have, what are we looking for now? I think y'all can get this, so I'm going to give y'all a second.
2 PQ?
Yeah, exactly. So we're heterozygous as a carrier because that's going to be this. And so heterozygous means you have 1 dominant allele and 1 recessive allele. And so if we know the recessive, and now we want to find this, we're going to have to go through a few steps, because we can't just subtract this because we don't know PQ yet, or P squared yet. So at this point, we know that this equals Q squared equals 0.04. So what we have to do here, anybody want to guess what we have to do here? that could get us going the right direction?
Take the square root.
Square.
Yeah. So if we take the square root of both sides, we end up with Q equals 0.2, which was our, which represents our gene frequency, or how many alleles in this population are little c. So now that we have Q, what would our next step be?
Plug that into the.
Yeah, right. So we plug it in over here. So I'll do it down here. P plus 0.2 equals 1. And so P is going to equal what?
Point eight.
Yeah, 0.8. Because 0.8 plus 0.2 equals 1. So now we know P. Now it's home free from here. So we know P and we know Q, so what's the next step we do?
2PQ.
Yeah. So now we get 2PQ up here. Isaac, you're pretty good at math, aren't you? He's catching on quick. So P, or 2, I don't know why stuff's not coming out the way what I wanted to say. P is 0.8, and Q is what? 0.2. And so now, just like we saw earlier, 2 times 0.16, and then 2 times 0.16 gives us 0.32. And so what percent of the population are heterozygous? Yeah, 32%. So 0.32 times 100 equals 32%. On the answers, as far as the homework, if you put 0.32, decimal or percentage is fine. So you don't have to be anal about which one's which. I just want to make sure you can follow it. All right, so that was good. So that's like a real world situation. Let me pass these out right now, and we might go over one of them, just to kind of get you a head start. So this will be worth 30 cents. And I did the midterm grades before class, and even though a lot of people had issues with the test, there's enough of stuff like this that if you get all your work in and all your labs and all that, then the grades really didn't turn out too bad at all. So if you can do fairly well on the test and then get all your other points, then you can probably get the grade that you're looking for. Let's see. That first one should be pretty easy, considering that's the same, decimal we used. And then the third one is similar to, and we'll stop right there. But that way you can see how that goes. So we'll do a different problem. All right, so number 2 says if 81% of a population is homozygous recessive for a given trait, we're not even telling you what it is, but we don't really need to know that. So if 81% of the population is homozygous recessive for a given trait, what is that telling us out of all these 2 equations? What is that giving us? Yeah, Q squared, just like the last one we went over. So that one tells us that 0.81%, or 81%, so that's 0.81 is Q squared. And now we want to know, instead of heterozygous, we want to know how many are homozygous dominant. So what's our first step here with this information?
Find and put the square root.
Yeah, take the square root of it. Yeah, so if we have q squared, and we need to find out this, we need to find out what this is first, right? So let's take the square root of q squared to get q. And so q, I believe, equals 0.09. No, that is 0.9, sorry. And what told me that was I was thinking to have two in a row that are, and that's what you can do to kind of catch it if you're not going to go and use your calculator for every little thing, you're just kind of going in a hurry, is I'm thinking, what's the chances of me getting two of the recessive alleles in a row, and that's 81%, then to just get one of them in a row, I have a better chance of that. So it's going to be higher than 0.09. It's going to be 9, which is 90%. So if you understand that concept, it'll help you make little mistakes like that. Keep you from making little mistakes like that. So now that we know Q, what is P? 0.1. Everybody got that? Okay. So P equals 0.1. So that's 10% of this population has the dominant allele. And so how do we get homozygous dominant from that? Square it, yeah. So PQ equals 0.1 squared, and PQ equals, make sure I write it down here. Yeah, 0.01. Which is smaller, so that makes sense. The chances of getting one dominant allele out of this bucket, it's greater when you don't have very many, just to get one versus two. So if you have just 10% in there, you don't have a very good chance of getting two in a row. So, so that's good. So from there, you know, if you're trying to find heterozygous, that's pretty straightforward. Anybody have any questions? You follow that pretty good? All right. So, all right. I'll just leave it up there. All right. Now we'll go back to the slides. There's only 2 pages in the book devoted to this, but I think it helps you understand a lot of other things. So now we're going to look at how we use this, and hopefully now that you understand kind of the flow of the equations, you can look at it. Let me give your mind a break. Let me see. So my son has this channel, and I put some of my daughter's workout videos up. Once you, just like 1 minute, but give you a break. And I'll tell you the exact way the test question kind of presents itself. So, we'll bring this up. This is some kind of little population of ferret-looking things. And I think it's looking at maybe fur color. So dominant is brown fur, and then the recessive is the lighter fur color. And so you have some heterozygotes, and you have some homozygous recessive, and you have some homozygous dominant. And basically, we're talking about evolution, and basically the Hardy-Weinberg equation is this baseline that we can use to see how much evolution is happening, because all the conditions for Hardy-Weinberg to be effective is that there's no evolution happening. So any deviation from that lets you know how fast the population is changing. So the allele frequencies always change. So how many dominants we have for something, how many recessive, these are always changing. But when we talk about the Hardy-Weinberg equilibrium, we're just in a pretend world that they're not changing. So this is the unlikely situation in which allele frequencies do not change between generations. So this is a hypothetical situation. It's not real. And here's all the 5 rules that, like the crash course told us earlier, the 5 rules that have to be in effect for Hardy-Weinberg to work out. One is that natural selection does not occur. So I want y'all to know we're going to talk about natural selection a lot, but with evolution, if you talk about the definition of what evolution is, it's random mutation, or natural selection acting on random mutation. And that's basically what the next couple chapters are about, is you have natural selection working on random mutations. So number 2 is mutations. That's where, when we're looking at DNA, maybe one of those A's switches to a T, or maybe one of those G's switches to an A, or any kind of situation like that. So majority of the time when one of those switches happen, it's bad for the organism, or it might be neutral. But every once in a while, that mutation may be beneficial. And so if it's a beneficial one, the natural selection is going to act on it. If it's a bad one, natural selection is going to act on it. It's going to weed it out. If it's a good one, natural selection is going to act on it and make it more prevalent. That allele will show up a lot more in this population of weird ferrets, because that particular ferret maybe has some kind of advantage that helps it to live longer, so it's going to live up to reproductive age. If it lives up to reproductive age, it's going to have more offspring, and so if it has more offspring, more of its alleles are going to spread through this population. Y'all follow that? So, but we'll elaborate on that concept a lot. If you understand that concept, you understand the drivers of what evolution is. So, so we're talking about Hardy-Weinberg, so we're saying that no mutations happen, no A's end up switching to T's, or G's to A's, or anything like that, and that natural selection doesn't pick out one allele and either weed it out or make it stronger in a population. And in these populations, you have to have really big populations, because it kind of dulls out any kind of random changes in alleles. So if you hit, anytime you have a lot of something, little changes one way or the other don't make a big difference. And that individuals made at random. So like they said, the surfers don't apply to this situation. And then there's no migration. So you're not having any, like, say you have someone in the United States and someone immigrates from China, and that brings a whole new, a lot of different alleles into that population. So you're not having any immigration, and you're not having any immigration. Immigration and immigration. When it starts with an E, that means that it's going out from the population. So you're not having any gene flow of people coming in or people going out. It's just staying static. That population staying static. So that aspect right there that I just said is relates to a test question. So you break the rules of the Hardy-Weinberg if individuals immigrate or emigrate, which means they leave. So that would break one of the rules of Hardy-Weinberg. So Hardy-Weinberg equilibrium will not occur in a population if you break any of these rules. So that's one of the test questions there. Basically, another test question is just being able to know that, I'm not going to tell you which one, but one of these three, you need to know either that P square represents homozygous dominant, or that 2PQ represents heterozygous, or that homozygous recessive represents Q squared. You just need to be able to associate those with what's what. I'm going to slide ahead to that slide, which is 35. So, well, we'll just kind of go through the slides. But so that's two questions. You just need to be able to associate which one goes with what. Also on slide 30, which is this one, basically, for Hardy-Weinberg equilibrium to take effect, you can't have any change. So evolution has changed. So what we're saying is that all allele frequencies stay the same. So evolution happens in a population, and so if you're not having any alleles changing, then Hardy-Weinberg equilibrium, all this math that we do will make sense and work. But if alleles are frequencies are changing at all, Hardy-Weinberg equilibrium will not occur. So it will occur if all the allele frequencies stay the same. Let's say that this kind of takes you back to, let me get up here. 1, 2. So, if you're looking at several generations in a population, and in one generation something disappears, and then you see it again in the second generation, which one of those three things does it have to be? Homozygous dominant, heterozygous, or homozygous recessive? There's only one that could apply to this. I heard a lot of heteros. It's a homo one. Recessive, yeah. Because think about this. Let's say that with cystic fibrosis we do a Punnett square. You have a carrier. Let's see. What am I doing? I didn't set this up very good. Sorry. It's been a while since we punted it up in here. Alright, so you have CC, you have a heterozygous here, you have a homozygous dominant, and you have a heterozygous. So in that first generation, generation 1, we don't have any phenotypes that have cystic fibrosis. And so now let's say that we take these two. In the next generation, we take those two and you have a homozygous dominant, heterozygous, and a homozygous recessive. Now, in the second generation, all of a sudden we have that phenotype again, and you only have a situation like that with homozygous recessive. So, so if you have some kind of population and a phenotype, this is the phenotype, is whether you have cystic fibrosis or not. You either have it or you don't. That's the phenotype. And let's say that it skips a generation and then it returns the second generation, then that's telling us that particular phenotype is homozygous recessive, because that's the only one it could be to be able to skip a generation. Y'all follow that? All right. So kind of save these as the review. I thought this might be better now that we're kind of digging into it to go ahead and address the questions. If you might be given a basic question, real basic question. So if it asks if it gives you like, for example, if it gives you what the dominant allele frequency is in a population, could you find the homozygous recessive or homozygous dominant? It'll be easy math where you can kind of do it in your head, but it'll make sure that you kind of know how to do this in your homework. If you can do the ones on your homework, that one will be easy. So, just kind of understanding that. All right. so let's kind of go back through this, and we'll kind of beat a dead horse a little bit, but for some of you, that'll help you out. So we know what the requirements are to have Hardy-Weinberg equilibrium, that there's basically no change in alleles happening. Assuming the assumptions of Hardy-Weinberg equilibrium are met, so basically that all these things are apply, then we have Hardy-Weinberg equilibrium, and these two equations will apply now. They'll represent the relationship between allele frequencies, this is allele frequencies, and genotype frequencies, this is genotype. So alleles where you just have one letter, genotypes where you're putting the mother and father alleles together, you're having two letters. So these are the genotypes, these are the genes or the alleles by themselves. So P is the frequency of the dominant allele, Q is the frequency of the recessive allele when you're talking about this equation. So looking at the little ferrets, since the D gene has two alleles, the frequency of the dominant allele plus the frequency of the recessive allele must equal 1. So we already kind of know that. If you know what the dominant is, you can find the recessive because it equals 1. P is the frequency of the dominant allele, Q is the recessive. So let's look at this. This is an allele from the mother, this is an allele from the mother, this is an allele from the father, this is an allele from the father. This is the genotype coming from the father, this is the genotype coming from the mother. And if you do a Punnett square, you can see how the Punnett square, because this is Mendelian genetics, Gregor Mendel, this is how his little pea plants worked. We're just applying that same concept to a big population. So now we can